Across subtropical groves, citrus trees carry round fruit filled with stored sunlight. Beneath each glossy rind, the fruit builds a layered chemical defense. In the white pith, one small molecule gathers with unmistakable bitterness. Its name is naringin, especially abundant in grapefruit and bitter orange. For the tree, that bitterness helps discourage hungry mouths and microbes. For people, it can turn brightness into a stubborn aftertaste. A glass may smell fresh, yet finish sharp, lingering and dry. The sweetness is present, but a molecular gate remains firmly closed. The locked gate is a sugar-decorated flavonoid, difficult to tame cleanly. Heat can push it apart, but heat also changes fruit character. Strong chemistry can remove bitterness, while taking more than bitterness away. Nature had already found a smaller, calmer answer in living soil. On fallen peel, fungi and bacteria encounter naringin as useful food. They do not overpower it, they recognize its vulnerable joins. Their enzymes approach the molecule like keys shaped by long seasons. Among those keys, one family would later be called Naringinase. The human story begins with 19th-century chemists studying bitter citrus extracts. In 1857, de Vries described naringin from grapefruit materials. The crystals carried a bitterness recognizable even in very small traces. Later chemists learned the molecule held sugars fastened to a plant core. They named the core Naringenin, part of the wider flavonoid world. Yet laboratory splitting often depended on acid, heat, and long patience. The living, cooler root remained hidden in mold, peel, and compost. By the 1930s, citrus science was becoming an industry science. Grapefruit juice promised refreshment, but bitterness limited its wider acceptance. In 1938, Hall reported an enzyme preparation that attacked naringin. The enzyme action came from fungal cultures, not harsh chemical treatment. Scientists named the helper naringinase, after the bitter compound it changed. This was a quiet turning point for the processing of citrus. Food researchers could now imagine debittering with biology, not brute force. Mid-century trials followed in juice rooms, pilot vessels, and laboratory glassware. Fungi such as Aspergillus became central sources for studying the enzyme. Grapefruit sections, juices, and peel extracts all entered the same story. The aim was simple: soften bitterness while honoring the fruit itself. At the molecular scale, naringinase is really a coordinated pair. One part removes a rhamnose sugar from the molecule's outer branch. The bitter molecule becomes prunin, a quieter halfway form. A second part then releases glucose from the remaining bond. What remains is naringenin, less intensely bitter and chemically useful. The enzyme does not smash the fruit's chemistry apart. It cradles one bond, invites water, and lets separation happen. Afterward, the enzyme is free to meet another molecule. Such patience is the deep economy of catalysis in living systems. Many tiny encounters can change the taste of a whole harvest. In juice, the harsh edge recedes without erasing citrus identity. In peel extracts, hidden value begins to move into practical reach. Some aroma molecules are also locked behind sugars inside plants. Related enzyme actions can release delicate notes in fruit and wine. Naringinase is best known for making grapefruit products taste gentler. It can help marmalades, juices, and extracts keep a better balance. It also helps turn citrus peel from residue into resource. Pressed rinds still contain pectin, oils, sugars, and valuable flavonoids. With enzyme treatment, bitter compounds can become more versatile ingredients. Naringenin is explored in food, cosmetic, and nutritional research pathways. The released sugars can support broader plans for citrus byproducts. In laboratories, naringinase becomes a tool for reading plant molecules. By removing sugars, it reveals how nature stores and protects compounds. That knowledge connects flavor, solubility, aroma, and plant defense. The enzyme also belongs to the larger story of fermentation. Selected microbes can grow on agricultural residues and produce useful proteins. In that circle, citrus waste may help answer citrus bitterness. Later designs held enzymes in place while liquids passed nearby. The helper could be kept while the transformed liquid moved onward. This idea shaped gentler processing across food and bio-based industries. Naringinase shows how precision can replace excess heat and chemicals. It works in water, near the conditions life already understands. Its history runs from bitter groves to old benches and modern biorefineries. Each chapter suggests a future where waste becomes raw material. Enzymes like this ask industry to borrow nature's patience. From stubborn rind to useful molecule, the path remains quietly green. Enzymes Bio began with a simple frustration we kept hearing. Sourcing many enzymes meant many suppliers and slow paperwork. We made it one place. Cleaner chemistry, ready for the future. Discover it at Enzymes Bio.